자, 음, 오늘 세미나 수업을 시작하도록 하겠습니다. 먼저 연자에 대한 소개를 좀 할게요. 오늘은 특별히 태국 방콕에서 티안신 일리암수완 교수님이 여러분들 세미나를 위해서 오늘 방문해 주셨고 교수님 그 이력을 간단하게 좀 설명을 드리면 어, 학부와 석사를 어, University of Karlsruhe 독일에서 학부랑 석사를 하셨고 박사는 스톡홀름 유니버시티 스웨덴에서 박사를 하셨습니다. 그리고 이 리서치 관심 있는 리서치 분야가 사실은 저희 쪽이랑 많이 다 있는데 몬테칼로 시뮬레이션인 레디에이션 테라피 앤 레디에이션 프로텍션 그리고 뉴트론 도시메트리, 마이크로 도시메트리 앤 레디에이션 인듀스드 DNA 데미지 모델링 그리고 predictive modeling of radiation induced normal tissue toxicity. 그래서 오늘 귀한 분을 모셨으니까 여러분들 세미나 잘 듣고 도움이 많이 있으면 좋겠습니다. 큰 박수로 맞이해 주시기 바랍니다.
Yes, thank you very much, Professor Min, for the invitation and for the introduction. So my name is Thiansin Liamsuwan from Chulalongkorn Royal Academy. So maybe Professor Min have already mentioned about my background. So I actually have background in physics in my bachelor and master degrees from University of Karlsruhe in Germany. And then later I moved to medical physics. So I did my PhD in medical radiation physics. And my PhD thesis is about development of Monte Carlo track structure simulations for protons and carbon ions in water under the supervision of Professor Hushan Michu at the Radiation Biophysics group in the medical school in Sweden, Karolinska Institutet. And later I came back to Thailand, so I worked as a nuclear scientist in Thailand Institute of Nuclear Technology for a couple of years, and then I moved to CRE or to become a lecturer in medical physics. So I'm now responsible for both master and PhD in medical physics and medical engineering. So as Professor Min has mentioned, my research interests, so my core background and my interest in application of Monte Carlo simulations in radiation therapy and radiation protection. And lately, I'm also interested in radiation risk assessment and microdosimetry and radiation dosimetry has been my interest since my PhD era, and also multi-scale modeling of radiation-induced biological effect, which is the topic of my talk today. So when I talk about multi-scale modeling, I mean mechanistic modeling of biological effects. So if you are familiar with radiation science, you know that radiation has effects on our biological targets, starting from DNA, cells to tissues. So to understand this process, we need to know the mechanism of this process in detail, and you can use mathematics and physics to understand this process in biology. So as we are, so my group consists mainly of physicists, right? So we use Monte Carlo simulations as a tools to model these kind of processes. We use a few Monte Carlo codes for this kind of works. For macroscopic calculation, like a conventional radiation dosimetry, we use like FITS and topaz. This pointer, right? Is this a pointer? Is it? It's okay. I just use my mouse. Okay, so for conventional radiation dosimetry, we use FITS and TOPAZ, and I heard that many of you also use TOPAZ here in Yonsei. And for very small size target, when we are interested in microscopic targets like DNA or cells, we opted to use Geon4 DNA. And most of our works are connected to treatment planning in the clinics in radiotherapy. So we usually use clinical treatment planning systems to produce treatment plans, right? But for modalities that is not available at our centers, we also use MATRAD, which is a research treatment planning system developed in DKFZ in Germany. And for secondary cancer risk assessment in radiation therapy, we also use computational phantoms, and I think many of you are familiar because you are also quite specialized here in Yonsei. So talking about biological response to ionizing radiation, it is multi-scale by nature. It covers physics, chemistry, and biology. It's span from nanoseconds to hours, days, and years if you talk about cancer induction. So therefore, to understand these kind of processes, we have to start from physics, and we have to explore in terms of chemistry and biology of this kind of response to really reach to the point of cell response. So the outline of my talk today will cover Monte Carlo tract structure simulation of water radiolysis, and I will touch a little bit about on microdosimetry, and most of the slides, most of the pages will be dedicated to radiobiological modeling, starting from DNA damage, DNA repair, and cell survival. So you may know about Monte Carlo simulations, and many of you are also using Monte Carlo simulation, right? It is a tool that we use to simulate radiation interactions. So it's based on the concept of random sampling of interaction characteristic based on probability density functions. So in fact, we use Monte Carlo simulation to simulate phase spaces of radiation in regions of interest. And we may classify Monte Carlo simulation as condensed history Monte Carlo and Monte Carlo tract structure simulation. So condensed history Monte Carlo group several interactions within specified path lengths. So it uses stopping power to characterize average energy loss of radiation over a given distance, and also use multiple scattering theories to characterize the scattering angle after the radiation passes a specific distance in the medium. So usually this kind of simulation is okay for conventional radiation dosimetry. So if the resolution is like hundreds of micrometer, you are fine with this kind of simulation. So on the other hand, Monte Carlo tract structure simulation simulates every interaction event by event, so every detail. That means the resolution is as good as the radiation mean free path, which is in atomic and molecular scales, so nanometer scales. So if you want to know energy deposition or dose in nanometer size target, then you cannot use the condensed history Monte Carlo, but you have to use Monte Carlo tract structure simulation to run your calculation. So thinking about our biological targets, what do we have? We have DNAs, we have chromatin fibers, we have chromosomes, cells, and tissues. And many of these targets are very small. For example, DNA has diameter of 2 to 3 nanometers. Chromatin fiber has a diameter of 30 nanometer, and cells have a diameter of 10 to 20 micrometers. So you can see that if you want to really know dose in this kind of targets, you have to use Monte Carlo tract structure simulation for your calculation. So like in conventional Monte Carlo simulation, the first thing when you use simulation is you want to validate your calculation, your model, right? So in macroscopic Monte Carlo simulation, you can do radiation dosimetry experiments quite well with good accuracy. But when the target is very small, then it's quite challenging to measure dose in nanometer size target very accurately. So what you can do to prove that your Monte Carlo simulation is accurate enough to be used for your problems of interest. So one way is to use Monte Carlo simulation to calculate macroscopic quantities, for instance, linear energy transfer, LET, or stopping power. So you can use Monte Carlo simulation to simulate energy depositions in macroscopic size target and calculate the LET and compare it with established database like P-star or ICRU database that your physics behind the track structure simulation is okay to be used further. And microdosimetry is very related to Monte Carlo track structure simulation. So, you know, radiation dosimetry, right? It's a field that we use to characterize dose in some mediums of interest. And microdosimetry is also, we want to characterize dose, but in very, very small target, in microscopic target. So it is proposed to be a method to characterize the radiation quality at the microscopic scale. So microdosimetry is actually conceptualized long time ago, like the first ICRU reports in 1983. So is this conceptualized under the assumption that radiation effects are related to radiation track structure and the size and nature of biological targets? One of the most commonly used microdosimetric quantities is the linear energy, usually written with the symbol Y. So linear energy is defined as energy imparted in a target volume by a single event divided by the mean chord length of the volume. And by single events, it means an event means a radiation track that impart energy in a volume of interest. So it's not just a single energy deposition point, but it is a single track, right? So from this definition, you can see that the unit of this is, for example, kV per micron, which is similar to what kind of quantity? LET or stopping power, right? So in fact, linear energy is it's like a microscopic counterpart of linear energy, and it is a stochastic quantity. That means we can really derive the frequency distribution of the linear energy. So the frequency average linear energy is the first moment of this distribution, and the dose average linear energy is the second moment of this distribution. So you may wonder what's the difference between LET and linear energy. LET is defined as the average energy loss per unit path length, right? While linear energy is energy imparted in the volume of interest divided by the mean chord length. So imagine we have ion tracks. These are the same ion tracks. If we have a sampling target volume at the core of the ion track, the LET and the linear energy may not be that much different. But since ionizing radiation produce ionizations, right? So that means there are a lot of secondary electrons produced, even though you are dealing with high LET radiations like carbon ions. So that means if the sampling volume just sample the delta ray or secondary electron tracks, the linear energy will be largely different from the LET, which is a characteristic of the ion itself. So that means if you are interested in very small targets, then maybe you need to use microdosimetry instead of LET to characterize your radiation beam. And in fact, if we plot the track structure of ions, which has the same LET, but different ions, in this case from helium, carbon, silicon to iron ions, they have the same LET, but you can see from this picture that they have completely different radiation track structures. So, and I hope that this picture can convince you that if you are, if you really want to work on microscopic scale, then maybe LET is not sufficient, but you need to use radiation track structure or microdosimetry to characterize the radiation quality of different kinds of radiation. So microdosimetry, so unlike LET, LET is an average value. So unless you don't attenuate your radiation so much, you should get the same LET for different target sizes, right? But microdosimetry quantities is very sensitive to the size of the target. So for example, we have calculated those mean linear energy for different ions for 10 nanometer and 100 nanometer size targets, and we found that depending on the size, the dose mean linear energy is completely different. So that means if you work with microdosimetry, you always have to mention about the dimension of your target of interest. Also, when we compare the dose mean LET and the dose mean linear energy for a proton beam and a carbon ion beam, we may see similar trends of the LET and the dose mean linear energy, but the magnitude is completely different, especially for sparsely ionizing radiation like protons. And also in terms of size dependency, we can see that the smaller the sampling target volume is, the larger difference is between the LET and the linear energy. So as I presented in one of the first slides, physics is very occurs in a very short period, like in femtosecond after the radiation is exposing to cells or DNAs. Later, it's like chemistry and biology that play a very important role into the induction of biological response to ionizing radiation. So to completely understand the process from physics to biology, we have to use like mechanistic models to explain all these steps. So one of the aim in radiation biophysics field is to link energy deposition or interaction of radiation to the cellular or tissue response. And this is really still a challenging task. We may characterize the radiation track structure quite well. We may model DNA damage and repair quite okay, but the link between DNA repair to like cell response or tissue response is still a big gap that is not well understood and still need to be explored. So in terms of biological response, first we have to explore what our cell is made of. So living cells are abundant with water, and ionizing radiations ionize and excite the water molecules, which initiate the subsequent water analysis. So we may classify the time stage of water radiolysis into different time scale. So the first femtosecond is just the physical state, just the physical interactions, where ionization introduce electron and water molecule ion in the system, and excitation initiate or produces excited water molecule. And after that, up to about 1 picosecond, the product from the physical stage will dissociate, thermalize, or decay and generate reactive chemical species. And after that, is only chemical reactions between these chemical species. So these species may diffuse and they may eventually react with each other. So all these steps need to be taken into account if you want to understand the biological response to ionizing radiation. So in fact, DNA is considered a sensitive target for ionizing radiation. If the damage to the DNA is induced by the physical energy deposition, we call it direct effect. If it's introduced or induced by reactive chemical species in the chemical stage, we call it indirect effect. And both direct and indirect effects are not negligible in both low and high LETs. So even though you work with carbon ions, you can still not neglect the indirect effect. So in terms of Monte Carlo simulation, Monte Carlo track structure simulation, we simulate the physical stage by simulating ionization and excitation mainly. And from ionization, we get water molecule ion and the sub-excitation electron. And from excitation, we have water molecule at different excitation stages. So this has to be included in the simulation. And then in the physical chemical stage, there's a chemical rearrangement of products of the physical stage in terms of dissociation, decay, and thermalization. So, and this chemical arrangement will produce reactive chemical species, either free radicals or molecular products. So the simulation code will use decay channels with branching ratios to simulate these kinds of chemical reactions. And then in the last stage, in the chemical stage, up to about 1 microsecond, the chemical species that are produced in the physical chemical stage will diffuse, right? And or if they may also react with each other and produce other products. So the diffusion is characterized by the diffusion coefficient, which is related to the root mean square descent of the chemical species. And the reaction between two species is characterized by the reaction radius, which is governed by the reaction rate and diffusion coefficients of both reacting species. So the Monte Carlo track structure simulation use a table of the diffusion coefficients and reaction rates to simulate the diffusion and reactions of these chemical species. And usually for radiobiology, we believe that only hydroxyl radicals are the major responsible species to cause indirect damage. So as you can see from this reaction table, we may get different kind of products from the chemical reactions, but in terms of biological effect modeling, we are interested only into hydroxyl radical written by OH in this table. This is a picture showing the time evolution of chemical species starting from a very dense track of alpha particle. So alpha particle is high LT radiation. And as you can see, when the time goes by, the spatial distribution of the chemical species is quite uniform. So that means even though you start with high LET, at sometimes when sometimes went by, you will get a uniform distribution of chemical species. So, and it also means that even though your target, your biological target is not hit by the direct effect, it may be get damaged by the indirect effect that is diffused to the further sites from the first interaction. So once we have the physics and chemistry ready, now it's time to think about how DNA is damaged by the radiation, either by direct or indirect effect. So we know that DNA is the sensitive target to ionizing radiation for both direct and indirect effect. There are two types of DNA damage. If the damage occurs in the sugar phosphate strand, it's called strand break. If it occurs in the volume of a base of the DNA, is called base damage. So strand break can be classified into single strand break and double strand break. Single strand break is isolated break in one DNA strand, and double strand break has at least two breaks in opposing strands that are close to each other. So in terms of biology, double strand break is more difficult to be repaired and considered to be more lethal. That means most likely to cause cell death than single strand break. So most of radiobiological modeling are focusing on double strand break induction and repair. So in the literature, there are different approaches to do DNA damage modeling. The detailed approach considers all stages of water radiolysis to model both direct and indirect effect. But there are also some simplified approaches that consider only physical interactions, but they use assumptions, either cluster analysis or threshold analysis, to model the DNA damage. And I will introduce you all these approaches in the coming slides. So for the detailed approach, you need to use Monte Carlo simulation, Monte Carlo track structure simulation to simulate the physical, chemical, and chemical stages of water radiolysis in a large working volume. And by large, I mean just micrometer. And then you use a small sampling volume in nanometer size target to represent DNA structures. So naturally, we want the DNAs to be hit by radiation in a random direction to mimic the radiation exposure to DNAs, right? So, and then once the geometry is set up, you can use a set of assumptions to model the DNA damage. For example, for direct effect, you can assume that if energy deposition in the physical stage is within an effective volume and larger than a threshold value, then the damage will be induced. While for the indirect effect, you can assume that the hydroxyl radical has a probability to induce DNA damage if it is located within a diffusion distance from the DNA. So this kind of assumptions vary between papers, between research groups. For example, in our group, we use a single threshold value of 17.5 electron volt, but some other group may use a varied probability from 5 to 35 electron volt. Also, the probability of indirect effect can also be varied between models. 0.13 has been proposed, and in our work, we use 0.4. So this depends on the calibration process of your model to the experimental data. So once you produce damage in the simulation, now it's time to classify the damage. As I said, we are mostly interested in double-strand break, but there are also single-strand break and base damage. So usually single-strand break is quite straightforwardly defined as a break in one strand, and double-strand break may be defined as two single-strand breaks on opposing strands, but within 10 base pairs from each other. So if you have more breaks inside a double-strand break, we call it complex double-strand break. And these complex damages are believed to be more difficult to be repaired and more related to the lethality of the cell. So for example, we have done calculation for protons of different energies and found that complex damage contributes around 10% of all DNA damage produced by protons. And as you can see, the contribution also increased with the decreased energy of protons. So that means with the increased LET. Kunrad and co-workers have also reported single-strand break and double-strand break use from detailed simulation for different kind of ions and plotted it against the LET. On the left is the single-strand break yields, on the right is double-strand break yields. So as you can see, the single-strand break decrease with the LET, but double-strand break tend to increase with the LET. And different ions with the same LET may produce different single-strand break and double-strand break yields, especially for the LET above 10 kV per micron. And these differences, if you see, there are three curves here, total, direct, and indirect. These differences are likely to come from the direct effect, which is related to the ion itself, not the chemical reactions. So another approach to model DNA damage is to use simplified method, such as clustering analysis. So in cluster analysis, you can you may only simulate physical track, only the physical stage of water radiolysis, and then you use clustering algorithms to group energy deposition in a volume of interest, and then relate the cluster size to specific types of DNA damage. So the most commonly used clustering algorithm is the density-based spatial clustering of application with noise, the so-called DB scan. So one of my students has also used DB scan cluster analysis to model DNA damage. And in that model, we simulate the physical interactions, we simulate the energy deposition event by event using Monte Carlo track structure simulation. And then we assume that is an energy deposition threshold for damage induction of 17.5 electron volt. And then the maximum distance between damage points to form a cluster is defined as the parameter epsilon. So, and this epsilon value is subject to calibration with experimental data. And a cluster is formed when at least 2 damages are present in the cluster. Otherwise, the isolated damage are considered as noises. So to calibrate our DB scan model, we assume that the ratio of double strand break to single strand break is equal to the cluster to noise ratio. And then we optimize the maximum radius for cluster formation, the epsilon value, with the reference double strand break to single strand break ratios. And then we have introduced a new metric for defining cluster density. We called it normalized quality of beam, which is defined as number of clusters per total energy deposited. And surprisingly, we found that this metric is has a strong linear relationship to the double strand break yields. So that leads us to the conclusion that if we use the normalized quality of beam as the reagent quality, at least tested for protons, we can just use a single conversion factor to calculate the double strand break yields. So this approach produced actually relatively good agreement to experimental data. And so it follows the same trends as experimental data and detailed simulation, but still we have to test this to this assumption for other particle types. And also probably we have to establish the conversion factors for the different cell lines. So the other simplified method is even more simplified. You just use a threshold value to relate energy deposition into DNA damage yield. So in the threshold analysis, you also simulate only physical tracks using Monte Carlo track structure simulation. It's actually similar to microdosimetry calculations. So you use small size target volume, usually in nanometer size, to sample energy depositions by radiation tracks. And then you relate the frequency of energy deposition larger than a threshold value to DNA damage yield. So just a single conversion factor to change, to convert this frequency of energy deposition to DNA damage yield. So this is, for example, one of our work that try to use the threshold analysis to model the DNA damage yield. So for example, when we calibrate this frequency of energy larger than epsilon deposit in the DNA size target, we found that if we set the threshold to 60 eV, 60 electron volt, then the multiplication, the product of this frequency and the number of targets per cell per unit mass can reproduce the single strand break yields very well, double strand break yields very well. And if we set the threshold value of 12 electron volt, our data reproduce the single strand break yields really well. So as you can see from this figure, the left-hand side show the single strand break yields and the right-hand side show the double strand break yields. So our calculation is in the black curve. And as you can see, with very simple relationship, we can really reproduce the experimental data. But you can also see that biological experiments have very large uncertainty. So it's not like in physics that you can measure those very accurately and precisely in biology. You talk about uncertainties in tens of percents or even higher. So DNA damage occurs maybe in the time scale of microsecond, which is very short. But after that, other biological processes comes into action. And one of the most important biological process, DNA repair. So usually when we look into the paper of modeling of DNA repair, most of the models focus on double-strand break repair because, as I mentioned earlier, it's believed to be the most toxic lesions from ionizing radiation. And in human cells, there are different pathways for double-strand break repairs. For example, homologous recombination, non-homologous enjoining, microhomology mediated enjoining or MMEJ, and single-strand annealing and SSA. So these pathways are actually cell cycle dependent. That means depending on the cell cycle stage, some pathway may be present, some may be not. So how we can model DNA repair? So DNA repair process actually a lot of biochemical reactions. That mean once you have double-strand break in your cell, a lot of proteins will be recruited to the breaks to do the repair. It's not one step process, a lot of steps process, right? So in latest work with one of my master students, we try to model all pathways that are available for human cells in HEJ, MMEJ, SSA, and HHR, and we come up with 29 steps of biochemical reactions. And if you have been to biochemistry class, I'm not sure, biochemical reactions, actually, you can convert the reaction into an ordinary differential equation. So it is actually characterized by reaction rate, usually written with the K and indexed with the step of interest, and the concentration of the protein complex, right? And Y represent the residual, the double-strand break with the protein complex. So that means if we introduce 29 biochemical reactions, we will get 29 ordinary differential equations. And as a physicist or engineers, you know that you can solve it numerically. And that's also what we do. We solve these equations numerically to get the reaction rates, a set of K values that really reproduce the experimental data of residual double-strand breaks after radiation exposure. These are example of our results. So from two experimental data, we have externally validated the model with other data. Now we only focus on protons, and actually we think that our model can reproduce the experimental data relatively well for protons of different energies and in different cell lines. So what's next after the double-strand break repair or DNA repair? So the final goal for biological effect modeling is, of course, to really link the physics to biological endpoints. And the endpoints of interest is, of course, cell survival, cell death, or if you are more into risk assessment, you may interested into cancer induction. But in this case, in our group, we are more into cell survival modeling, which is related to radiotherapy treatment planning. So how can you mechanistically link DNA damage, DNA repair with cell death? So there are several approaches. So conventionally, you can use phenomenological function like the LQ model. And I hope that you have heard about that. It's just an exponential function to describe cell survival, but it's phenomenological. Phenomenological means it doesn't give you the mechanism. It just gives you a fitting functions, which fits the experiment relatively well. But if you want to understand mechanism behind, then you need to have a mechanistic models. And one way of doing that, you can use models such as the modifying parameter reduced to lesions kinetics model proposed by Sakata and co-worker in 2023. So in this model, the parameters that are inputted in this model is the residual double-strand break used at different time after radiations. So with our double-strand break repair kinetics, we can get the use of double-strand breaks after specific time point, and we can input it into this equation and solve it and calibrate the parameters like beta, lambda, gamma, and eta with the experimental cell survival fraction. So these are examples of our results from mechanistic modeling of cell survival fractions. So also here we discriminate between the optimization set and the external validation set. So from one single experiment, we can reproduce the cell survival fractions relatively well, but only up to around 4 to 6 gray of proton radiations. But given in mind, this is a conventionally fractionated radiation therapy, so 4 to 6 gray is sufficient for predicting the cell survival fractions. So some limitations and maybe some future aspects that we are thinking about is to include the cell cycle dependence into the DNA repair kinetics. As I mentioned, DNA repair is quite dependent on cell cycle, but we haven't taken it into account yet. And also, as you can see, so in this picture, the blue curve is actually from the mechanistic model, and the red curve is the LQ model. So symbols are experimental data. As you can see at high dose, mechanistic model tends to underestimate the survival fraction. It means it kills cells more than it should be at high doses. So when we discuss this with the biologists, they say that maybe we should also include some damage tolerance mechanisms at high doses. It means that even though the cells or DNAs are damaged by ionizing radiation, but it still persists to survive. So this is something that we could not consider into our models, and it will be a future improvement of the model. So in a summary, I hope I can convince you that radiobiological modeling is inherently multi-scale, from nanometer scale track structure to macroscopic biological outcomes. And also it covers different time scales from femtoseconds, picoseconds, microseconds to days and years. And Monte Carlo track structure simulations provide the foundation for understanding energy deposition and water radiolysis in living cells. For DNA damage modeling, we can do it in detail, taking into account physics, chemistry, and biology, or we can use simplified approach like cluster analysis or threshold analysis. And we can link DNA damage and repair to cell death using mechanistic models, so not only phenomenological models. And mechanistic model integrated physics, chemistry, and biology have potential to improve predictions of radiation therapy and radiation risk assessment, at least in, I hope so. Okay, and with this, I thank for your attention. And if you have any questions, then feel free to ask.